Right, so we were talking about HIV virus. So let's talk about its replication cycle and its genomic variety, right? And replication cycle, let's suppose it enters into a human cell, right? Let me draw a human cell. It may be macrophage or it may be a T lymphocyte, right? And now we have to see first how virus enters, and once it enters, how it multiplies, replicates, and one virus enter and how millions of virus come out. You know how efficiently it replicates? A person who becomes HIV positive, every day he makes more than 1 billion virus. I mean, if you get HIV, you are billionaire as far as this virus is concerned, right? So this is one way to be billionaire. So it is so efficient in replication. And if we really want to control this disease, we have to hit the virus at weak points in its replication cycle. So we must know the replication, as doctor, we must know its replication cycle very well, and we must know that during its replication cycle, what are the vulnerable points where we can give the drugs and damage the replication cycle. So let's start with it. There will be little repetition, so just bear with me. So we make the virus again. By now you know the virus very well, it's almost your friend. And here is the, yes, what is this protein I'm drawing? Capsid. What is this P? P24. Rapidly, what is this? I just want to see. RNA dimer. Yes, what is it? Reverse transcriptase, integrase, protease. Very good. And what is this protein? Matrix protein. Excellent. And P17. Very good. I'm impressed. And what were these proteins? GP41. And what are the caps over them? The naughty caps? GP120. Right? What is it? GP120. Now we start. This virus is approaching our cell, and then what happens? Right? Let's suppose that I will make it a little bit out of proportion. This is your GP41. There's little repetition here, and this is what? 120. Very happy because the other cell is CD4 positive. It is its primary attachment, right? Then its other thing come out. What is the other thing? Another binding site to bind with which receptor? Chemokine receptor. Let's suppose that if it is macrophage, it is CCR5, right? So primary attachment, secondary attachment, right? Once this attachment is done, what is the next step? That this will shift on the side. So I'm going to make the viral protein over here now, and it is more near. Primary attachments still remain in the same, right? This is, I'm going to make the same, right? You understand what I'm doing? This is the same attachment complex. What is the important thing? That now this was what? GP41. When both of them are engaged, it will come out and hook over there, and then pull these membranes together. So next step will be that, yes, it is also lipid bilayer. Human cell also has lipid bilayer. And next step, when this will bring it together, then this membrane and the other membrane, they will fuse with each other, and at the fusion point, what will happen? That viral envelope lipid bilayer and human cell lipid membrane, they will fuse together, and this will become the part of our membrane, and out of this, this is the material, this is the thing which will jump in. Is that right? This structure will jump in. It means basically up to now what we have seen, there is attachment, primary, secondary, and fusion. Once fusion is done there, then this core protein has entered in, and this core protein with all its contents, you still remember, keep on telling me, RNA, yes, rest in peace. Reverse transcriptase, integrase, and protease, right? The next step is that our proteases which are in the cell, they will attack capsid protein. What is this protein? P24, yes. This will dissolve and these components will come out, right? Now they are in our cytoplasm, right? Now once this is in our cytoplasm, I'm going to make now one RNA over here. This one RNA I bring out. This is viral RNA and this is single stranded, right? These are single stranded positive polarity dimer. Now this virus RNA comes and the first enzyme which will become active and work on this is, what is this enzyme? Reverse transcriptase. What is this enzyme? Reverse transcriptase. Let me draw the reverse transcriptase properly. This is we're very happy because it can start at action. It has double action enzyme, right? And it is having, it has a glue bottle. It can, with this glue bottle, it can attach the nucleotide here and make complementary DNA. It will read the RNA and make DNA. So this point of the enzyme is called polymerase. But which polymerase? RNA dependent DNA polymerase. And so I will talk about this domain later. This is one enzyme with two functional domains. What is this domain? Polymerase domain. Now this polymerase domain, what it will do, it will read the RNA and make complementary DNA. And when complementary DNA is made, what are these? Nucleotides. You know what are these? Nucleotides. They are added, right? Complementary nucleotides are added. Am I clear? Now when these nucleotides are added, this DNA strand is made. Once this DNA strand is made, the next step comes that this is again RNA of virus. Is that right? This section will occur on both strands. It has two strands. Both strands will undergo these mechanisms. And now here it has made what thing? Complementary DNA strand. And now you know what happens. You know what happens? This was the original template from which complementary DNA, single-stranded complementary DNA is made. Now this point become active. Now this point become active. And what will it do? We'll look at it. What it's going to do. This is its polymerase point, and this will start cutting. It will start cutting the RNA. I mean, this is very, it's not a loyalty that it is going to for auto destruction. The template RNA which came, as soon as from this RNA it make first complementary DNA, right? This domain become active and it start destroying RNA, the template. Is that right? It's a bit of very low, cheap activity, but it happens even in virus, not only in humans. So what happened? This RNA is destroyed. Before the destruction of this thing, now it was a duplex, RNA DNA duplex. But when this domain of reverse transcriptase become active, it will destroy the template. When the template is destroyed, what is left there? Single-stranded DNA. Is that right? It was required, it was needed to destroy this. Why? Because then another enzyme will come and now it will read this DNA and make complementary DNA. What is going to happen? It will read the single-stranded DNA and make complementary DNA, and in this step, when RNA has been removed from here and this is left single, then its complementary DNA will be made, and now it has become double-stranded DNA. What it has become? Double-stranded DNA. Now this point, which destroys the RNA, it is called RNase H. So we can say reverse transcriptase enzyme is double action enzyme. Number one, it has RNA dependent DNA polymerase activity. Number two, it has RNase H activity. But it is very intelligent. First it make the copy and then it will destroy. Sometimes it may happen as stupid that it just RNA come and it destroy the RNA. If virus has such kind of stupid reverse transcriptase, this virus will not survive. Why? Because DNA will not be made. You are getting it? Is this clear? Now, what is happening that by this time we have made double-stranded DNA. Then this double-stranded DNA will be transferred to our nucleus, to our cell nucleus, right? This will be transferred to our cell nucleus, and suppose this is our nucleus, this is our nuclear membrane, right? Now this will be carried into our nucleus, right? And once it is carried in our nucleus, then suppose this is our double-stranded DNA, human double-stranded DNA, right? And this is also human double-stranded DNA. Now the next enzyme will come into action, which is called integrase. Very good. The next enzyme will come into action, which is called integrase. Now let me show how this enzyme works, integrase. Integrase enzyme is like this, that it is having two bottles, two polymerase, what is this? Sticking points, binder, and one cutter, and it has one cutter. Right. Now what it will do? Actually, by this cutter, it will cut our DNA. It will put a cut in our DNA. Our DNA is a double-stranded long DNA. It will put a cut there. You remember rest in peace, integrase enzyme? Now this integrase enzyme will produce a cut in our DNA and separate our DNA. And then, with the end of our DNA, fit the viral DNA. You are understanding? So what is happening, that let's suppose, okay, I will make a human DNA with different color, so viral DNA remains black. Let's suppose human DNA is, I'm making with the green color, right? Then it will make a cut in that, this enzyme, and then it will make, fit into it, what is this? Viral DNA, right? Of course, it's double-stranded viral DNA, which was present over here. Now it has come over here. Now this function, that cutting our DNA and making the viral DNA fit into that, I mean it will cut and then tie over here and stitch over here. Now viral DNA becomes the part of our DNA, right? Now this viral DNA is called proviral DNA. What we call it? Proviral DNA. This is also proviral DNA because eventually it can make new virus. So what do you call it? Proviral DNA. Is that right? Now this proviral DNA gets inserted at multiple points within our DNA, and it becomes integrated forever. It becomes the part of our DNA forever. Now there's an issue. The drugs which we are presently using, they can destroy the virus at multiple levels, but we don't have any drug up to now which can basically remove this DNA which has been integrated in our genomic material. That is why once you are HIV positive, unfortunately, presently medical knowledge says you will be always HIV positive, because virus put its DNA into our DNA, and until our cells are surviving, virus is surviving. Virus genetic material is only destroyed when we are destroyed. Am I clear? That is second reason that we are unable to handle this disease or cure this disease. Presently, the drugs which we are using, they suppress the, you can say, replication of the virus or re-entry of virus from one cell to the other cell, right? But we cannot remove this integrated piece, and that is the reason the virus is, in a way, a permanent resident of our genomic material. Is that right? It's a resident evil here, right? Now let's come back. I will talk what this genetic material is doing there. But before that, let's come over here, that we were talking about the drugs, right? I will continue with some drugs more, but just a little. You remember I told you there was a drug here? What was this? Yes? Mara? Viroc. Say it loudly. Mara? I don't hear. Mara? Very good. Right? This was CCR5 blocker. Then there was another drug which could basically destroy this. Very good. What is the name of this drug? Anfuvertide. This was fusion inhibitor. Is that right? Now we come to some drugs here. Yes, very interesting. We play a game with the reverse transcriptase enzyme. You know, humans like not only fool each other, they also fool the virus, right? We make a special game with the virus. You know what we do? You know what this virus is, this enzyme is doing? It is catching up the nucleotides. What are these? Nucleotide. And attaching the nucleotide with each other and making a DNA. Is that right? What is the structure of nucleotide? Let me tell you. Let's suppose this is the phosphate, this is the sugar unit. What is this? Sugar unit, and ribose or deoxyribose. And what is this? Nitrogenous base. Adenine, guanine, right? Now, this is, I'm showing you a nucleotide. Now remember, every nucleotide sugar, it is having a hydroxyl unit here. What is it? Hydroxyl unit here. Hydroxyl unit is like a hook. What these enzymes do, they catch one nucleotide. Let's suppose if here there is adenine, they will catch a nucleoside or nucleotide with adenine has what? Uracil, right? Or if here it is, you can say cytosine, what it will catch? Guanine, right? Now what happens? This nucleotide will be taken. This nucleotide will be taken and it will be inserted here, complementary, and every nucleotide should have this hydroxyl point. This is 3-end hydroxyl. Now what is the importance of this hydroxyl? This tail of nucleotide. Very good. That next nucleotide, when it will come, it will hook with this. Then next nucleotide will come, hook with this. You are understanding? These tails are very important. These are called hydroxyl tails or hydroxyl points in the nucleotide, right? Now how many nucleotides are there? There can be adenine here, or there may be guanine here, or uracil here, so on and so forth. Now we play a trick with the virus. Normally when virus is entering in our cytoplasm, it finds the complementary nucleotide and start making the chain. Start making the chain of viral DNA. What we do a fraud. What we do, we have made in the pharmaceutically a nucleotide. What is this nucleotide? Suppose with adenine, but it does not have a tail, but there is no tail. There is no hydroxyl point. You are understanding? We give this drug to the patient. If I am HIV positive, you give me a drug like this, nucleotides or nucleosides which do not have hydroxyl. If you give me this, they will go through my blood and then they will enter into cells. And then, while virus is making this chain, sometimes it will catch this drug. It will think it is the natural, normal nucleotide. And now this is not normal. I can say it is dangerous. It is, we have given to the patient to fool the virus reverse transcriptase. So sometimes if you give this drug to the patient, while this is making this copy, somewhere it may catch and put the drug. But it doesn't have a tail. Now can it make chain further? No. You are understanding? Chain will be stopped here. So we have many drugs like this. What are these drugs? These are nucleotides and nucleosides. What are these? Nucleosides and nucleotides. So these are nucleosides and nucleotides. These are pseudo. They are not true, right? They are given pharmacologically. And reverse transcriptase catches these molecules, put in the chain, and then it catches the next but cannot attach. And DNA cannot be made. Do you think virus cycle will go forward? No. Reverse transcriptase has been foolen. I think foolen or what? Huh? My English is strange. Fooled, yes. Actually, I learned English from a man who speaks multiple languages. So due to that reason, it is not foolen, it is really fooled, right? I feel fooled myself. Okay. So what happened that this is fooled by pharmacologically administered nucleosides and nucleotides which do not have tails, which do not have 3 hydroxyls. And once these molecules are inserted here, then what happens? Chain termination occurs, right? So such drugs are called, yes, such drugs are called nucleoside or nucleotide reverse transcriptase inhibitors. What are these drugs? nucleoside, nucleotide, reverse transcriptase inhibitor. There are many drugs in this group. You can know only 2, 3. That will be enough. For example, have you heard of didanosine? You have not heard of didanosine. Okay, have you heard of lamivudine? Yes. Okay, lamivudine. Have you heard of, yes, what else is zudovudine? Yes. All of them are these fraudulent molecules, right? We are fooling the virus, right? So these drugs are called, what are these drugs? Nucleoside, nucleotide, reverse transcriptase inhibitor, right? They inhibit the action because once it put a fraudulent molecule here, then it cannot work further, right? So it's out of work. So we say these molecules are NRTIs, right? Nucleoside, nucleotide, reverse transcriptase inhibitor. So what I will write the name here? Lamivudine, didanosine, yes, zidovudine, and there are many other also, right? So these drugs, so what we can do, you can give a drug, attachment fail with co-receptor, you can give a drug, fusion does not occur, but if attachment occur, fusion occur, it enters, you fool the reverse transcriptase, provide it with the pseudo molecules, and it put in the chain, and then chain termination occur. Am I clear? Then we came up with another drug, right? That drug is very funny. Let's suppose we imagine another drug which comes and which blocks, this is the polymerase point, right? What is this drug has done? It has made the polymerase point dysfunctional, right? This drug is different than providing the fraudulent molecule, because these molecules were going and enzyme remain functioning, but provided with the nucleoside, nucleotide, which are put in the chain but do not have hydroxyl point, so chain cannot continue. Then someone came up with a drug that is making the polymerase point dysfunctional. This will also inhibit the function of reverse transcriptase. This drug will also inhibit the function of reverse transcriptase. But tell me one thing, is it a nucleotide, nucleoside molecule? No. So what should be the group of this drug? These are non-nucleotide, nucleoside, reverse transcriptase inhibitor. So we can call it NNRTIs. What we call it? NNRTIs. Right? Now you can imagine how these two drugs are different. NRTIs are pseudonucleotide without hydroxyl point, right? Enzyme is fooled and put them into chain, and then because there is no hydroxyl, next nucleotide cannot be added and chain termination occur. And NNRTIs, non-nucleotide nucleoside reverse transcriptase inhibitors, are not pseudonucleotide. They are basically molecules which directly bind with the polymerase and make it dysfunctional. Can you tell me the single drug in this group? Just tell me one drug in this group. Don't forget we are going to be doctor or doctors. Just one drug, not many. Nevirapine. What is the drug here? Nevirapine. Right? So how many groups of drugs we have done? Number one is attachment inhibitor, Maraviroc, CCR5 inhibitor. Second was fusion inhibitor. What was that? Enfuvirtide. Third group came NRTIs. What is this? Like lamivudine, didanosine, zidovudine. Then there are NNRTIs, non-nucleotide nucleoside reverse transcriptase inhibitor, which make the polymerase molecule dysfunctional. One classical example is nevirapine. Any question up to this? You have understood? Yes.
So these NRTIs that we have, do they affect the other DNA replication taking place in other cells or in body?
Yeah, this is a very interesting question. She is saying that if we give the patient these pseudo molecules, will it affect our normal physiological DNA polymerization? Yes, it can. But what we do, we try to use those molecules as antiretroviral drug, which are little bit different, right? and molecules which have more affinity for viral reverse transcriptase inhibitor and less affinity for human DNA polymerase molecules. Is that right? But anyway, many of these side effects occur because our own biological mechanisms are also affected and suppressed. For example, bone marrow failure, right? Because in bone marrow the cells are very rapidly multiplying and they are making a lot of DNA, and if we provide pseudo molecules, sometimes they fit into our normal physiological bone marrow function replicative pool, and bone marrow failure may occur. We'll talk in detail when I will teach you a full lecture about antiretroviral drugs. This is just discussion, right? Okay, so any more question? Okay, let's move forward. What is the next point to hit? Hit this man. What is this man? Integrase enzyme. If you give a drug and destroy this, and it cannot integrate viral DNA into our system, virus does not. Now listen very carefully. This proviral DNA, this is also called cDNA, complementary DNA. This is cDNA, proviral DNA. It cannot make the virus until it is not integrated in our DNA. Or if we give a drug which will disturb this enzyme, what is this enzyme? Integrase. If we give a drug, then integrase fails, and integrase cannot integrate viral DNA here. Do you think virus can replicate very well? No. So we have one drug here also. All the money on it and effort on research against HIV has not been wasted. Now they are promising results. So there's a drug here. Which drug is here? Yes. Okay, before you tell me something funny, let me tell you what is the drug. The drug which inhibits integrase, that is called Raltegravir. Raltegravir. VIR. Raltegravir, right? So Raltegravir is the drug, one example of the drug which belong to the group of integrase inhibitors. So up to now, how many groups? CCR5 inhibitors, GP41 fusion inhibitors, NRTIs inhibitors, which are pseudonucleotide type things, then NNRTIs, which make the polymerase dysfunctional, and if virus passes through all these stages, we make a trouble here. Don't let it integrate. And drug is? Say it loudly. I feel your silencer in your mouth. Yes? Raltegravir, right? So this is how virus is, proviral DNA is integrated there. Once this become a permanent resident evil of our genomic material, now here it will sit, and one day it become active and start making a lot of copies. Right? Now, why it should make copy? Before it start functioning and making viral RNA and viral proteins, and one virus entered and now millions of viruses going